The main themes of microbiology. By the end of this lecture, you will be able to answer the questions: what is microbiology and why we study microbes? You'll also be able to distinguish between beneficial and harmful microbes. Based on their individual focus, I will discuss a few of the career options available to microbiologists as well. You'll learn some general characteristics that define microbes, and then you'll learn about some of the great contributions to the field of microbiology and the scientists who made those contributions. Lastly, I will briefly discuss how microbes are classified, named, and their place on the evolutionary tree of life. Microbiology is the study of organisms that are too small to be seen with the naked eye and must be viewed with a microscope. Later, you will see just how small they are in relation to macroscopic organisms that can be seen with the naked eye. Microorganisms, also known as microbes, are either living or non-living. The first 5 listed here are considered living microbes because they are all made of cells, the basic unit of life. Bacteria and archaea look similar, but they are very different from bacteria in many ways, including their metabolic capabilities and the makeup of their cell wall and cell membrane. Fungi can be multicellular, made of many cells like mushrooms, or unicellular, made of a single cell like yeasts. Protists are an informal group that include organisms that don't really fit into any other category. This group includes protozoa, which are very diverse and interesting organisms, as well as algae. Algae are considered protists, but there are also some bacteria in this group, like cyanobacteria, which were previously thought to be algae and later discovered that they are actually bacteria. Helminths, also known as worms, are not microbes, but we study them because their microscopic eggs and larvae are associated with human diseases. We can also group organisms by their cell type. Bacteria and archaea are prokaryotic cells, versus fungi, protists, and helminths, which are composed of eukaryotic cells. Prokaryotic cells are much simpler and about 10 times smaller than a typical eukaryotic cell. Viruses are considered non-living because they are not made up of cells. They are simply genetic material in the form of DNA or RNA surrounded by a sac of protein. Viruses can infect all types of cells, and yes, even bacterial cells. Viruses need the help of a living cell in order to multiply. Once inside of a host cell, the virus takes over the machinery, so the cell spends its energy producing more viruses. We study microbes because they are ubiquitous, which means they are found everywhere. There are microbes that live in places that other organisms cannot survive, like extreme heat, extreme cold, extremely salty conditions, and extremely high pressure conditions, just to name a few. They are naturally found in us, on us, and all around us. Those microbes that live in and on us are called normal microbiota. These microbes help keep us healthy, but only if they stay in the places where they normally reside in or on our bodies. For example, we all have E. coli at the end of our digestive tract, but if we don't wash our hands after we use the restroom and before eating, there is a chance that the E. coli can be swallowed and end up in the intestines where they are not normally found. This can cause diarrhea and other problems. This leads me to pathogens, which are microbes that cause disease. These microbes can be normal microbes that are out of their normal place, or other types of microbes that are not part of our normal microbiota that can cause disease in humans as well. Pathogenic microbes can also cause diseases in animals and in plants, which can be a big problem for farmers of food animals like cows and agricultural crops like rice and corn. We also study microbes because they can help us in food production. During their metabolism, they create byproducts that are very helpful in making bread, beer, wine, vinegar, yogurt, cheese, and the delicious drink kombucha. They are also responsible for production of antibiotics and pharmaceutical drugs, vaccines, amino acids, vitamins, and organic solvents like ethanol and acetone. Microbes help our environment by degrading dead animals and plants, which can then serve as nutrients for plants and other organisms. Microbes provide plants with the nitrogen needed for growth, and other microbes are used to clean up our environment by converting toxic substances to less harmful forms. For example, there is an archaea that converts oil to methane, which is useful for cleaning up oil spills in large bodies of water. We call this bioremediation. The majority of the known microbes are good and are generally harmless, with the exception of those organisms that I previously mentioned, normal microbiota that can get out of place. These good bacteria are beneficial to us or the environment. The bad microbes are pathogens which cause disease. These diseases are either emerging, newly discovered diseases like COVID-19 or the monkeypox, or re-emerging diseases, old ones that continue to come back and cause issues like the flu, cholera, and smallpox. Microbiology is a large field within biology with many subfields within itself and therefore many career options. The public health microbiologist is responsible for monitoring and controlling the spread of disease in communities. They track, control, and try to prevent the spread of diseases and other health hazards. The environmental microbiologists study the ways in which microorganisms interact with the natural environment. They may study environmental process of microbes in air, water, soils, and sediments. Biotechnology is a fast-growing field of microbiology where biotechnologists use microbes to produce desired products. For example, microbes have been used to produce human insulin for diabetics. This insulin is less harmful than in the past when insulin was extracted from animals. Agricultural microbiologists study plants and animals as well as the beneficial interactions they have with microbes. This chart shows the sizes of various microorganisms that can only be viewed with a microscope and macroscopic objects that can be seen without a microscope. Objects must be about 100 to 200 micrometers to be seen with the naked eye. A typical eukaryotic animal cell is microscopic and measures about 10 micrometers in diameter. A typical bacterial cell is 1 micrometer in diameter. That's 10 times smaller than the animal cell. And viruses are even smaller, about 10 times smaller than a bacterial cell. The bright field microscope, also known as light microscopes that we use in lab, allow us to see objects at about 100 nanometers or 0.1 micrometers or larger. Anything smaller than 100 nanometers requires a very powerful electron microscope. You should know the general size ranges of different types of organisms and cellular components and the equipment used to view them. This is a handy chart that will also help give you an idea of how much smaller some objects are compared to others mentioned on the previous slide. The 6 types of microbes that I introduced earlier can be grouped into 3 categories based on the types of cells they're made of. Prokaryotic cells are very small and simplistic. These organisms are unicellular, which means the entire organism consists of one single cell, as opposed to multicellular organisms like plants and animals that consist of many cells and many different cell types. Prokaryotic cells do not have a membrane-bound nucleus, or even a nucleus for that matter, and they do not have membrane-bound organelles. As mentioned previously, bacteria and archaea are prokaryotes. Viruses are about 10 times smaller than prokaryotes, and they are considered acellular because they are not made up of cells. They are simply nucleic acid, DNA, or RNA surrounded by a protein coat. Eukaryotes encompass all other microbes, the protists, fungi, and helminths. Eukaryotic cells are much smaller and more complex than prokaryotes. Some eukaryotes are unicellular like protozoa, but there are others that are multicellular like algae and fungi. Eukaryotic cells contain a membrane-bound nucleus that encloses the DNA and organelles that are surrounded by membranes. Prokaryotic cells do not have these internal cellular structures. Looking back in the history, the theory of evolution suggests that the first prokaryotes appeared some 3.8 billion years ago, and eukaryotes showed up a little later, about 1.8 billion years ago. It is thought that prokaryotes evolved into eukaryotic cells over the course of time. This is known as the endosymbiotic theory. Endo means inside, and symbiosis is when two organisms live in close contact of each other, and this interaction offers an advantage to both. Endosymbiosis is when one organism lives inside of another and both benefit. It is thought that early proto-eukaryotic cells engulfed photosynthetic bacteria and aerobic bacteria. These bacteria formed an endosymbiotic relationship with the proto-eukaryotic cell and eventually became what we call organelles. The photosynthetic bacteria became known as chloroplasts, and the aerobic bacteria are what we now know as the mitochondria. You can read about this in section 3.2 of the online text. If it were not for thousands of scientists contributing to the field of microbiology, our life expectancy would be very short. We can now expect to live past 40 years old because microbiologists discovered that certain diseases and infections are caused by microorganisms, and other scientists discovered that microbes produce antibiotics that can treat the infections. Our food is kept cold because scientists discovered that microbes cause food spoilage. But our food is also more tasty because of the fermentation process of microbes. We can't talk about all of the scientists that contributed to the field of microbiology, but we'll discuss a few here. Antony van Leeuwenhoek, who designed one of the first microscopes, Edward Jenner, who used the scientific method to produce the smallpox vaccine, Louis Pasteur, who disproved the theory of spontaneous generation, and others who made great advances in medical microbiology and invented microbiology lab techniques. Antony van Leeuwenhoek was a Dutch linen merchant who wanted to take a closer look at the quality of his textiles. So he created a single lens microscope that was able to magnify objects about 300 times. This crude handheld microscope was made up of a lens, a specimen holder, a focus screw, and a handle to hold the microscope up to your eye. The ability to magnify such small objects piqued his curiosity, and he began to examine insects and other things like rainwater and plaque scrapings from teeth. In these samples, he observed tiny microorganisms which he called animalcules and drew pictures of his observations. These drawings were very accurate and are very similar to the organisms we can view with the modern light microscope. These discoveries opened a whole new microscopic world and the field of microbiology. The first step of the scientific method is making observations about something in the natural world. We notice patterns of behavior or trends and are curious as to why they happen. We collect information about our observations and use that information to ask questions that can be answered about the natural phenomena by doing some experiments. The questions begin with why, what, when, and how. For example, why isn't the remote working? Are the batteries dead? Is the remote broken? Once you have several questions to answer, then you form a hypothesis. The hypothesis is a statement of what you think the answer to your question might be and includes a prediction that you will test. A hypothesis should be falsifiable, meaning it should be able to be proven wrong based on another scientist's experiments and interpretation of the results. A hypothesis generally has the format of if, then, because. For example, if the batteries are dead in the remote control, then the remote will work when the batteries are replaced because the remote needs power to function. Next, we make predictions based on our hypothesis. For example, if we replace the batteries in the remote, it will work. We then conduct experiments to test our hypotheses. Experiments can take many years or several minutes depending on what you're studying. The results of your experiments are observed and/or measured. These results should be recorded for later analysis and will either support or refute your hypothesis. If your research is complete, then you can draw conclusions. For example, if I replace the batteries in the remote and it starts working again, the results support my hypothesis. But if I replace the batteries and the remote doesn't work, the results refute my hypothesis, and I'd need to go back and possibly ask more questions and conduct more experiments. When scientists finish a research project, the results of their study is reported in scientific journals. Their work is then peer-reviewed. This means their work is verified by other expert scientists in their field before the results are published. Once published, their work is open season for other scientists to falsify or to perform more experiments to gain a deeper understanding about that specific natural phenomena. In summary, the scientific method is a systematic way to investigate and answer questions the scientist formulates about the natural world. Scientists make an observation while collecting initial data, then ask questions they want to answer based on their observations. Next, a falsifiable hypothesis is formulated and written in the if, then, because format. Predictions are made based on what the scientist expects will happen, and then experiments are designed to test the predictions. After experiments are performed, data is collected and analyzed. Conclusions are drawn, and the scientist decides whether the results support or refute the hypothesis. In real life, scientists repeat experiments several times to make sure the results are repeatable before reporting their data to the scientific community. I'm sure you've heard of theories and laws. You should understand the difference between the two. A theory is what one or more hypotheses become once they've been verified several times over and over, accepted by the scientific community to be true. Much research has gone into proving various aspects of a phenomena or several related phenomenon so that the information as a whole provides a deeper understanding. A law is a statement of fact. It is generally taken to be true. Look up some examples of theories and laws. Also, watch this video for a better understanding of a scientific law versus a scientific theory. Edward Jenner used the scientific method to develop the smallpox vaccine. He observed that cows had a form of pox that are similar to smallpox. He also observed that milkmaids acquired these cowpox on their hands from milking the cows, and those milkmaids were immune from getting smallpox. Jenner hypothesized that the cowpox was closely related to smallpox and could possibly be used on people to give them some type of protection from smallpox, similar to the protection the milkmaids with cowpox had acquired. To test this hypothesis, he took scrapings from the cowpox blisters on the hands of the milkmaids and inoculated the pus into a boy who had never been exposed to smallpox. The boy developed very minor symptoms but generally remained healthy. A few weeks later, the child was exposed twice to the pus from a smallpox lesion, but did not acquire smallpox and was deemed immune. Jenner went on to inoculate 23 other test subjects with cowpox pus and also used pus from one child to inoculate another. All of the test subjects acquired protection from the smallpox. Jenner published his results and his detailed experiments. Other local physicians began to use this method of vaccination. Over the next 100 years, many scientists used Jenner's methods to develop vaccines against other human pathogens, and artificial immunity became a known and well-established theory. In 1979, cowpox was declared eradicated due to the early works of Edward Jenner. Long ago, people believed in spontaneous generation, that living things could spontaneously arise from vital forces present in non-living things or decomposing matter, like maggots appearing on rotten meat. This is because they could not see the microscopic organisms with their eyes that eventually developed into larger macroscopic organisms. Louis Pasteur disproved this theory and replaced it with the theory of biogenesis, that living things can only arise from other living things. In one experiment, he filled 2 swan-necked flasks with broth and applied heat to sterilize it. That is, to kill any living cells that may have been in the broth. He broke the neck at the top of one of the flasks and left the neck of the other flask intact. The flask with the broken neck was exposed to the air and therefore any airborne microbes that were able to enter the broth and grow. The broth in the other flask remained sterile because microbes were unable to travel up the swan neck and enter the broth. Louis Pasteur also contributed to the germ theory of disease along with Robert Koch and John Snow. People used to believe that diseases were caused by bad character, committing sins, or due to poverty even. But it was eventually discovered that diseases can be caused by microscopic organisms. Pasteur was a very busy scientist. He also showed that microbes cause fermentation of sugars that can result in food spoilage, which is why it's important to refrigerate perishables. But sometimes fermentation makes food and certain drinks taste very good and provides healthful benefits. Pasteur also developed the process of pasteurization to prolong the shelf life of foods and drinks. Robert Koch established Koch's postulates, a sequence of experimental steps that was used to verify that a specific microbe caused a specific disease. He used his postulates to identify the microorganisms that cause anthrax, tuberculosis, and cholera. In addition, he also developed pure culture methods to isolate a pure culture from a mixed culture. Watch this video to see how scientists transformed the way we think about disease. Once it was established that microorganisms cause disease, there was also a need for techniques to prevent the spread of pathogenic microbes. Aseptic techniques were developed when it was discovered that the human body is a source of infection. Dr. Oliver Wendell Holmes observed that surprisingly, mothers of home births had fewer infections than those who gave birth in hospitals. Dr. Semmelweis correlated infections acquired in the hospital with doctors who went directly from the autopsy room to the maternity ward without washing their hands. Their bodies were the source of infection, transporting pathogenic organisms from the corpses to the women giving birth. Joseph Lister developed aseptic techniques to reduce the number of microbes in medical settings and prevent wound infections. He discovered that by simply disinfecting hands with chemicals prior to surgery or delivery of babies, healthcare workers could prevent infection. He also developed aseptic techniques to sterilize surgical and other medical equipment, again, to prevent the transfer of pathogens. There are thousands of microorganisms. Taxonomy is a system for organizing, classifying, and naming living things, originally created by Carl von Linné. Taxonomy involves classification, arranging organisms into groups. Nomenclature is a systematic method to assign names to organisms, and identification, which involves determining and recording traits of organisms for placement into taxonomic groups with similar characteristics. The levels of classification are as follows: domain, kingdom, phylum for animal kingdom, or division for the plant kingdom, class, order, family, genus, and species. You can use the handy mnemonic, did King Peter or David come over for good spaghetti, to remember the order of the levels of classification. You should remember those. We simply refer to organisms by their genus and species name. For example, Homo sapiens. Homo is the genus and sapien is the species. Here is a sample taxonomy of Homo sapiens, also known as humans. We belong to the domain Eukarya, the Animalia kingdom, the phylum Chordata. Recall phylum is used for organisms in the animal kingdom. The class Mammalia, the order Primates, the family Hominoidea, the genus Homo, and the species Sapiens. Notice how the members of each taxonomic level decrease until there is only one specific organism. Names of microbes and other organisms have a binomial or scientific nomenclature, which just means we refer to each microorganism by two names, the genus and the species names. The genus name is always capitalized, and the species name is always lowercase. You should be aware that when typing the name of organisms, the genus and species name should always be italicized. When writing a report, the first time the organism name is introduced, it should be written out with the abbreviated name in parentheses. In this case, Staphylococcus aureus is typed in italics the first time it's introduced in a report, and when abbreviated, the first letter of the genus name is capitalized, and the species name is typed out and both are italicized. You should type out the names of organisms correctly in your work. When writing the names of organisms by hand, the genus name is capitalized and the species name is lowercase. To signify that it is the name of an organism, it is underlined. When handwritten, it is abbreviated in the same manner as previously described, but must also be underlined. The largest taxonomic level of life is the domain, and there are three: bacteria, archaea, and eukarya. All living things are classified into one of these three domains as represented on this phylogenetic tree of life. The yellow circle at the bottom of the tree represents an ancestral organism from which all other organisms evolved. The bacteria domain contains all bacteria and are the true bacteria. They are called true bacteria because archaea are similar in appearance to bacteria, but they are not true bacteria. Microorganisms in the domain archaea are adapted to extreme environments, and therefore some of the molecules that make up their cellular structures are very different from true bacteria. Organisms in the bacteria and archaea domains are made up of prokaryotic cells. The domain eukarya contains all other organisms. Organisms in this domain are made up of eukaryotic cells. You will notice that viruses are not found on the phylogenetic tree of life. This is because viruses are acellular and therefore not made of cells and therefore not alive. This is the end of the main themes of microbiology. Thank you for listening.